thank you for the invitation. This is the title of my talk, and I'm going to begin with the evolution of the electron microscope. Now I can't advance my slides. Why is that? Yes, I can. So when I began in the early 1960s, image analysis consisted more or less of, can you see the entire top of my screen? Because I have a bar across that.
Yeah, we could see it.
Okay,
Image
fine.
analysis.
No, there's stuff in front of mine. Anyway, consisted of guessing at what you thought your structure looked like and building a sort of simple like ping pong ball model of what it seemed to be like. Well, now, John Finch and Aaron Klug built a model of the human wart virus and compared it in great detail, as you can see here, to actual electron micrographs of the virus in negative stain. They, in order to prove that this model was unique and not just that it sort of seemed to be correct, they tilted the virus particle in the electron microscope, and they tilted the model by the same amount and about the same axis to show there was good agreement. You can see a sort of single eye here, is what Aaron called it, and this ring surrounding it, and this sort of threefold view here, and the same over here. Anyway, that was at the time the method of structure determination is. That is, you build a model, compare the model to the images, and then you tilt to prove the uniqueness. And of course, if the model didn't agree, then you had to refine the model and again, do the comparison. This was not very satisfactory. But Aaron Klug and I realized that an electron micrograph is a projection of a structure, and that if you tilted in this case, the beam or the structure, you could collect projections. And the Fourier transform of each projection was a central section through the three-dimensional Fourier transform. So you could build up the Fourier transform in 3 dimensions and then back transform it to get the image. And we applied it in a first instance to the T4 virus phage tail. And this was a resolution of about 35 angstroms. Then, so now we had a method, a general method for solving structures without having to guess at them. With Tony Crowther, Linda Amos, and John Finch, we generated the first 2 single particle reconstructions. One was tomato bushy stunt virus, and we combined these 3 images indicated by the arrows to produce the 3-dimensional map. Of course, we are helped by the fact that this has a Cauchy-Drille symmetry. Of course, for every view that you had, you got 60 views. And also human wart virus with only 2 views and a single tilt. So these 2 images that you see here were from a single tilt pair, and it was enough with a Cauchy-Drille symmetry to generate a 3-dimensional structure. So at this point, we had molecular resolution by electron microscopy. Now, the key at the time was to use negative stain, which inherently limits the resolution you can achieve. Uranium salts are good negative stains because they scatter quants strongly, providing contrast, amplitude contrast in the image. And so here's a sort of plot of the number of electrons. So when the electrons go through the structure, they are not scattered as strongly as by the stain. So you get this kind of distribution of electron density. The resolution is limited because the stain occurs as clumps about 1 nanometer in size. So if you think about clumps of stain, here's a clump of stain, and what you're looking at in the electron micrograph is a hole in the stain. And of course, the hole is only as well defined as the size of the, about the size of the clump. So you can see you would miss the finer details of the structure. And in fact, negative stain limits you to maybe at best 1.5 or 2 nanometers. Now, Unwin and Henderson, Richard Henderson and Nigel Unwin, replaced negative stain the glucose, which protected the in this case, two-dimensional crystals of the bacteriorhodopsin, protected it from the vacuum of the microscope. So this is their image, which you can see looks kind of featureless. But if you take a diffraction pattern of this image, you can see you get very nicely resolved crystalline reflections out to about 7 angstroms. So even though you can't see it because of the density of the glucose, you can still obtain structure. And in fact, they tilted this structure and produced the first three-dimensional image. They tilted, much as I showed you in the Drozier-Klug image of tilting the crystals about an axis, and collected data. And each of these columns is an alpha helix. This was also the first three-dimensional map of a membrane protein at about 7 angstroms. So it was an all alpha structure. Now, glucose is not good embedding agent for single particles because the particles are hidden by the equal density glucose layer. So at the time, a lot of people went to work on two-dimensional crystals because they could solve them in the EM, but they couldn't do single particle work. Now, ice is a better embedding agent than glucose because it's less dense. So here's an image from Bob Glaser and Ken Taylor. They built a cold state for their microscope, and they were able to obtain an image of a catalase, thin catalase crystal. And the diffraction pattern in the image goes out to about 1.5 nanometers. So the question was a preservation only as good as negative stain, about 1.5 nanometers? And they took electron diffraction patterns of these same samples, and they could in fact see reflections out to near atomic resolution. So the problem was not the preservation in these crystals, but in fact it had to do with the microscope itself. Well, the good news was that frozen water embedding makes particles visible. You can see particles here. It's a frozen hydrated picture. The trouble is that there's ice. Here's bend contours due to ice particles. So the bad news was that ice crystals alter the contrast and they disrupt structures. So if you wanted to study things like cells or something, you would be in some trouble. Well, along came Jacques Dubochet and his colleagues and found out that by plunge freezing into liquid ethane, you could produce vitreous ice, which at the time was considered to be impossible. And he had a good deal of trouble getting his work on vitreous ice published, but he did. But when this picture appeared in Nature, everyone immediately took up the use of vitreous ice. About the same time this picture came out, there was also an article by Nigel Unwin on his acetylcholine receptor. Anyway, now we had a way to embed particles. Now, the thing you should know about in the electron microscope is that in focus, there is no contrast. So in order to get the contrast you see here, you have to actually defocus the microscope to produce defocus phase contrast. Now, Aaron Klug and Harold Erickson had earlier worked out the way in which defocusing produces phase contrast. So here is what we call a contrast transfer function, showing this is a reciprocal space measure. This is resolution this way. This is the amplitude for those Fourier coefficients. And in this region, you get correct contrast. In this region where it crosses zero, you get no contrast, no information. And here the contrast is reversed. So Erickson and Kluge showed how image contrast is generated by defocus, how it is altered, and how to correct for it. During this period, Joachim Frank had decided that the future lay in work on single particles, because it was what the electron microscope was best at. It could see single particles, unlike X-ray diffraction, excuse me, or NMR. So here was his first attempt. He had developed this package called Spider. And here was his first attempt by taking a bunch of images in one orientation, aligning them and averaging to produce an average and a rotationally averaged image. Now, this next step is, to me, one of the key steps in single particle work. And that is the introduction of multivariate statistics by Joachim and Maren van Heel. And basically, what you can do is align, and using multivariate statistics, you can classify particles, and you can sort them, the particles, by orientation, conformation, and composition. And you can produce these class averages out of a field of particles shown here. This is a very important step. It is used today. It is always the beginning place for any structural study that I can think of when you have a field of single particles. Now, you'll be hearing from Fred Sigworth, but Fred introduced maximum likelihood. And in this case, the particles, rather than being grouped into particle being put into one bin and not into another, in maximum likelihood, you put particles into every class average, but weighted by the probability that they belong to that class. If you don't do that, here's the structure you're trying to get the average of, and here's your reference structure. The problem is you have reference bias, so that even as you try and extract this information from a field of structures with noise, you wind up eventually just getting back your initial reference. But if you use maximum likelihood, you avoid this pit. And even though it takes many repeats, you eventually get to the right structure. So this was an important addition that is used, and it prevents this iterative alignment from bias and from falling into local minima. So I consider this an important step in image analysis, one which is used heavily in all the programs I know about. Now, when I began with Aaron Klug, this is what we dealt with. This is an IBM 1790. All of our displays were on line printer by overprinting to produce density and by encoding amplitudes and phases, and then circling lines of constant density. And you can see over here when we began up till around the year 2020, there was a 10 to the 12-fold increase in the computing power, and this has made an enormous difference. And there have been many improvements in software since the MRC software package in 1970 and Joachim Frank's single particle Spider software. There are many packages. I'm not going to detail them. You'll probably hear about some of them and may use many of them singly or in series in your work. There have also been improvements to the electron microscope. Here's the original microscope built by Ernst Ruska in 1933. Here's the sort of microscope that John Finch used to produce our images that Aaron and I and John used. And here's, of course, the cryos. The price has gone up quite significantly. Anyway, one of the advances are multiple grid stable cryo-EM stages. This is very important. Higher voltage, which is good for thicker specimens, but probably not for very thin ones. Field emission guns made it in an instant change in resolutions that people could achieve from about 7 angstroms, as Hunman Henderson had done, to about 4, but we were still stuck until we had direct electron detectors, which further improved resolution to 2 angstroms and better. And energy filters, which remove inelastically scattered electrons, thus improving contrast. Now, here is an example of a movie made with a direct electron detector. And these are virus particles, you can see. And this is a run through the 40 frames. Well, it's pretty hard to tell what's going on. So in order to see about this problem that had plagued us for years, the EM community, it was called beam-induced motion. That is, when you turn on the electron beam, things move. So to better see this, here we have an average 10 of those frames of the 40 frames that were being recorded. And if you look at these particles, you can see they actually move considerably, although down here in the corner, they don't. You can see this one doesn't move, whereas this one moves quite a bit. So the problem with that had plagued everyone was that while they were taking their 1-second exposure, the particles were moving. And people called images where the motion was very small, they called those particles shiny because they seemed to be shiny. Anyway, now that you've been able to divide a single exposure up into 40 frames, you can correct for the motion. And in fact, this is what Nico Grigoriev and Bridget Carragher and their colleagues did. And of course, here's if you don't correct for the motion. And here, if you do, you get these nice shiny particles. So the direct electron detector all but eliminated the loss of resolution due to beam-induced motion. Well, here's a sort of example of what can be done in the resolution revolution. And here's a 1.25 angstrom map of ferritin done by the Holger Stark lab. And you can see here's 2D classification. Here is realigning, and here is a refinement of the defocus values. Here is polishing, meaning correcting for the beam-induced motion. And again, you go through cycles and cycles and cycles of this, sorting particles, combining them, refining their orientation, polishing them, until finally you can get this kind of map on apoferritin. And here is a high threshold cutoff, which shows that you can actually detect atoms. These little red things are, of course, the atoms in the selected amino acids. And here is the lower cutoff to show you what people are more conventionally seeing. It's absolutely stunning. And of course, has turned structural biology on its ear. I want to now spend a few minutes talking about a tomography. They remember in the original article that Aaron Kluge and I wrote that you can collect tilted images, which are projections, and you can combine them, and then you can use that information to generate a 3-dimensional map. So that is the basis of tomography. Now, we recall that the electron beam, you really is very destructive. And I sort of thought, I'll speak for me personally, that you couldn't really collect a tilt series because you could take one image, but then the electron beam would have destroyed your sample. But Wolfgang Baumeister showed that you could indeed collect a tilt series by constraining the dose, and you could generate a three-dimensional structure, a three-dimensional tomogram of your structure. And here is a slice. He used just lipid vesicles to show that it was possible. And I must say, it was quite stunning when he showed that. And of course, people began to use this tomography on frozen hydrated samples in order to study cells. And there was an additional feature that he introduced, and that was subtomogram averaging. So in single particle work, you collect two-dimensional images and you sort them into classes. In subtomogram averaging, what you do is you cut out three-dimensional images, like you'd cut out the image of this nuclear pore somewhere in here, cut it in 3 dimensions, and then you classify them, you sort them, use multivariate statistics, just as you do for single two-dimensional single particles, in order to generate classes and produce class averages. So you can look at the structure of the nuclear pore complex. So today, using subtomogram averaging from tomograms, the best resolution to date is about 3 angstroms, but more typically, it's not as good as that. But it is still the way to study your structure in situ in a cell, and to look at the variations in structure and composition. Now, the initial excitement about using generating cryotomograms was that the problem is the cell body is too thick to get through the electron beam. So people were confined to thin specimens that can get through the electron beam. For example, Filopodia or Lamellipodia, or bacteria that weren't too big. But in Mike Marco, here shown with his cat, introduced focused ion beam milling, in which you can actually use a beam of gallium ions to thin the sample and make it thin enough to get the electron beam through. And here's some of his early examples of a bacterium that he had thinned. And here is his sort of tomogram in which he had segmented it to show the features. Anyway, this meant that you were no longer constrained to the thin parts of cells, but that you could in fact thin them with this focused ion beam milling. And sort of the general procedure, here's the device here, and here's the gun that produces the beam of ions that mills away, blasts away the sample. And in general, here's your cell poking above the grid surface. And what you do is with a focused ion beam, you produce a thin lamella that's held by the sides of the cell. And then you can, this is thin enough, maybe a quarter of a nanometer, a quarter of a micron, 250 nanometers, and you can then tilt it about this axis and collect a tomogram. So now you were freed, you could actually look at the interiors of cells. Now, given that you can get the beautiful tomograms, such as the one shown here, how do you find structures? Medali et al. In this paper introduced template matching. So they would derive templates from the databases and then use them to, using cross-correlation, to pick out the structures of interest. And then what they could do is replace those located structures with colored versions of themselves. So here's an example of a segmented tomogram. You can see filaments and ribosomes and membranes. So here's one example to show you what can be done with this. Here's an example of the workflow in which you find your cell. This is the LRRK2 protein associated with Parkinson's disease. And here is, you can mill the sample. You can look at it with fluorescence microscopy. You can then collect a tomogram. You can do subtomogram averaging, and then you can do model building to produce a structure. And here is a slice through the tomogram. This is the LARC2 protein raptor and microtubules. You can then generate subtomogram averages among the classes, and then you can use the volume you have done and modeling in order to produce what was the first three-dimensional structure for this protein done in situ on microtubules by these groups of people. Now, I had originally called deficiencies of electron microscopy, but these days people don't like deficiencies. They prefer to call them challenges. So I've now switched it to challenges. So the electron beam is damaging, and you can control this by controlling the dose. Under focus, phase contrast, and astigmatism, which affects amplitudes and phases. We've talked about that. Digitization of the image affects amplitudes and phases. Beam-induced motion limits resolution. We've talked about that. Interpolation in the image, which we haven't talked about, reduces high resolution interpolation, but these can all be controlled. Image distortion by the lenses. These are taken into account by some of the programs. Beam tilt, which alters phases. Insufficient depth of field, which alters amplitudes and phases. These are the things you have to be worrying about, or the programs have to be worrying about as you process data. Lack of parallel illumination alters the phases. Now, here are some things that we might not be able to control, but we should be aware of. Multiple scattering within the sample, which alters amplitudes and phases. Inelastically scattered electrons, which add to the background. And of course, the energy filter helps with that. And lack of coherence of the electron beam, which limits resolution. Now, thin specimens, of course, help with multiple scattering. They inelastically scattered electrons, the energy filter, of course, and they now have more coherent electron beams, which are needed if you want to go down into the region below about 2 angstroms. Challenges to tomography. Well, the first obvious challenge is that you can't tilt by 180 degrees. You can tilt from -60 to +60, but you're missing the wedge of data in the Fourier transform from 60 to 120. It's called the missing wedge. And also the incremental step in between each tilt gives rise to missing wedges between the slices. The milling beam damages the outer layers of the lamella. Very nice work by Bronwyn Lucas and Nico Grigoriev to show that there is some damage into about 50 nanometers into the lamella. And you're limited to a small volume of the cell. Serial sectioning is not possible, as you saw in this sort of cartoon. So you can't always, you can't see the whole cell. Milling and imaging is slow and can be difficult. And there's always a chance that you will mill somewhere and the structure you're interested in is not near lamella, always a possibility. And it's also possible that lamella are lost when you're moving between the FibSem machine to the electron microscope. So these are some of the challenges in doing them. And of course, there's always ice contamination that affects all sorts of production of frozen hydrated specimens. So I want to talk finally about the promise, and I don't want to talk about all the aspects of it. I just want to mention a few. Now, the laser faceplate, which is still in the experimental stages, provides almost perfect stable phase contrast. So there's no need to defocus. And this will allow us to obtain near atomic resolution of even smaller structures than the one was mentioned by Lee Kuo. the development of clonable tags that are easily recognized by template matching in images and tomograms. So this is the EM equivalent to a fluorescent tag. It'll help you locate structures in the tomogram and be able to segment them. This is still a development, so we're hoping that that will pan out well. And finally, artificial intelligence or machine learning aided image analysis and particle picking, clustering, segmenting, and more, still being developed and tested by many labs around the world. And then the combined use of super resolution cryolight microscopy with cryo-electron tomography, which give us more precise locations of our target molecules and aid in milling. And I finally want to end by showing you something that despite all of these challenges and so on, the results are pretty spectacular. So here is a movie going through the sections of a tomogram from the VIA lab. And here it is being segmented. So you can see it being replaced. And then it's being tilted. So you can see the stunning image detail that's available in tomograms. You can see membranes, you can see the nucleus, which is here, the nuclear membrane. Here's the inside of the nucleus. Here's a mitochondrion. Here are filaments, ribosomes, and so on. It's really quite exciting. And I want to end by saying that Bob Glazer quotes us, we overestimate will be done in the short term. That's when you're rated grant. You always overestimate what you can get done and underestimate what will be done in the long term. And I must say, having been at the beginning of this business, I never dreamed that today we'd see atoms with the electron microscope, atoms of biological specimens. So thank you. I apologize that I cannot remain for the question and answer session, but I will be happy if anyone wants to email me. I'd be happy to answer their questions.
So Miguel says, any adverse effects on the sample caused by uranium salts, structurally speaking, that is to say?
Actually, yeah, some of the salts work at different pHs. So some of them, like uranyl acetate, are quite acidic, and you can get a more neutral form. So that is something you can observe. There's also on occasion some positive staining that goes on with it. And also it's very interesting, but uranyl acetate actually fixes samples. So there was a time study done in which the negative stain was introduced at various time points and was able to fix and freeze the sample. So I'm sure there could be negative effects as well, although by and large over the years I've seen that the envelope that you get from negative stain almost always looks very similar to the final structure done by a cryo-EM. And I will also add that negative stain is a very good place to begin when you're beginning any structure and you want to see how good your sample is. Negative stain is very good. Doesn't mean your ice will be good, but negative stain is good. It's easy to do, and it's very informative. Excuse me.
Okay, I think we'll take this one final question in the chat and then we'll let you go. This question is, what is your opinion on the application of high voltage EM on thick samples like 1000 kV?
Yeah, there used to be million volt microscopes. They weren't as elegant as the ones today, but the penetration doesn't go up as rapidly as a voltage does. So you do get more penetration. But of course, I imagine that the problem of dealing with a microscope increases, and it would be increasingly expensive to do so. So I think there are some around, but I think for the most part, the biological community has stuck to 300 and not pushed for million volt microscopes, but they definitely would allow you to look at thicker samples, but not, let's say, 3 times as thick, maybe only 1.5 times as thick compared to 300 volts.
Okay. Thank you again. Thank you for taking the time to join us. Always a pleasure and much appreciated.
My pleasure.